So that's a 3' overhang, not a 5' overhang. So you got those two options, the 5' overhang that's skewed this way, the 3' overhang that's skewed that way. And then there are a few that do cut in the middle. And those leave what we call a blunt end. See, there's nothing hanging off here. And so these do not, the blunt end ones don't automatically come back together with ends cut by the same enzyme, like the other two did. These can come back together with any other blunt end cutter, whether it's the same enzyme or a different one. But just like if you've ever worked in a wood shop, you know, if I take a 2x4 and I cut it straight across and I want to butt end join it to another 2x4, that's a weak joint, okay? That's a hard joint to keep together. It would be a lot better if I just broke it, right? And I had jagged ends and I put those back together. Now there's a lot of interacting surface area, right? That's a strong joint if I glue it back together. But a straight blunt end cut, I glue that back together, that's a weak joint. This is a weak joint compared to that. Now in practice, the sugar phosphate bonding is the same in both cases. But energetically speaking, it's harder to glue this back together than it is this one. Sorry, that's what you need. The take-home point is there are 3 kinds of ends these things do, okay? And you need to stare at this long enough to understand what makes a 3' overhang and what makes a 5' overhang and what makes a blunt. All right. Practically speaking, though, you cut any piece of DNA with enzyme X and any other piece of DNA with enzyme X, and you can put those pieces together. When you do put them together, there's 2 steps that have to happen. The base pairs have to hydrogen bond, right? And that happens automatically. You don't need an enzyme to do it. They're just gonna, at the right temperature and salt conditions, they're just gonna base pair again. But the sugar phosphate backbones do require an enzyme to repair their covalent bonds. And that enzyme is called ligase. We're going to see DNA ligase again when we talk about DNA replication. Okay, so you need something to glue the sugar phosphate backbones together, and it requires this enzyme to make that happen. All right. Oh, yeah, here you go again. EcoR1 is an E. coli enzyme. It leaves a 5' overhang. Small one is a blunt cutter. KBN1 is a 3' end overhang. And when you work in this business, sort of get used to, you just automatically learn a few of these guys. This is a picture of DNA electrophoresis that I just drew by hand up here. Okay, so there's a slab gel with wells. You load the DNA in there. You're going to do this if you're in the lab, by the way. And it's in this big box, which has fluid buffer in it to conduct electricity and keep the pH the same. And it's got these electrodes on the ends. The DNA always migrates toward the plus electrode if it's neutral pH. What's to say about that? I don't think so for now. For class purposes, the point of electrophoresis in our context is to separate things by size. In this case, the length of the DNA fragment. You can do electrophoresis on proteins. That's a little more complicated, but it again separates them by size. That separation is not linear in relation to size. It is logarithmic in relation to size. So this is the smallest band in the mixture. That's the biggest band in the mixture. But it's not like I drew it here where I approximated equal distance between the bands and they're each 1,000 bases different. In reality, it would be something like this, where you've got the 3,000, which by the way, we would say 3 kilobases, 3,000 bases. Here would be the 2,000, but the 1,000 wouldn't be down here at an equal distance. It would be at a longer distance. So they compress as the size gets larger. Practically speaking, why do smaller fragments migrate more quickly? Well, if you could shrink yourself down to,